Okay, you guys ready to get going today? Alright, so we got a lot to cover. Buckle up. We're going to move pretty quick because I'm going to assume that everybody's kept up with the assignments and has already familiarized themselves with the basic ideas that we're going to be talking today, particularly about gross neuroanatomy of the nervous system that we will pick up from where we left last class. Quick reminder, as far as I'm aware, you guys are welcome to correct me, but I believe next Thursday is our first exam. Be prepared, bring your laptops or whatever smart tablet or phone you need to interface with Blackboard learning system that we will use to take the exam. Okay? No supplemental materials or websites or anything like that will be permitted nor tolerated, and so come prepared. We're going to cover a lot of material today and next class in preparation for that exam. So come prepared with that information already read in terms of the assignment, but dial in this weekend, make sure you're studying. This is a lot of material. The brain is complicated, and we're going to be tested on a lot of material that we'll be covering. Okay, so fair warning, it is manageable and attainable for everyone to succeed though. Okay, I'm going to do my best to help you get there. So I'm going to move quick today. If I've had too much coffee, gong me or slow me down. I'm going to rely on your feedback and you guys to keep me at a pace that is amenable. Does that sound like a good deal?
Yes.
Okay. Yes, ma'am.
Is there still going to be like online quizzes that you post online?
Yeah, well, there are online quizzes as far as I'm aware that are already associated with the textbook.
Oh, okay.
So that's in the syllabus, and you should be able to access that and interact with that. But I will try to make some sample ones on Blackboard, probably over the weekend, once I have a chance to decompress over this large cup of coffee that I pounded for this morning. It's breakfast. Does that sound like a good deal too? We're all on the same page? Everybody good to go? All right, let's do this thing. So we're going to talk about gross neuroanatomy today. Basically, the primary features and elements of the nervous system. Okay, so we're going to categorize the nervous system in a couple different ways, and then we're going to take a closer peek into some of the features of these nervous systems, particularly some of the main features or structures that we'll be referring to for the rest of the quarter. Okay, so we can categorize it in two main ways. What we mean by gross in this reference isn't disgusting. It is general, broad level neuroanatomy. At this level, we can categorize the central nervous system versus the peripheral nervous system. By central nervous system, we basically mean the brain and spinal cord, and by periphery, we mean those nerves in the arms, legs, and rest of the body. Okay, so we can see that visualized here. The central nervous system shown in blue. The author's done a very convenient trick to help us by coloring the central nervous system blue, and you see the whole spinal cord going all the way down, and everything here in yellow. All that crazy stuff are all our peripheral nerves representing the peripheral nervous system. And in fact, there's a whole lot more that are way too tiny to even draw. But we already did our reductionism and levels of analysis journey last class, so we'll leave it there. But there's a whole lot more, and same thing for the blue-colored central nervous system. Okay, there's a lot more, but that's just encapsulated within those colors, right? Billions of neurons within each of these systems. But this is basic conceptualization, all right? Now, as you might imagine, this is our way of taxonomizing. By taxonomy, what we mean is not stuffing jackalopes up on the wall, okay, for any of you taxidermists in the room, okay? But what we mean is basically a way of categorizing something. So, okay, so this is how we can categorize things, but they both interface and interact as sort of a unitary, even broader system, the way that sort of nature has designed it. Okay, so again, this is our central nervous system. We have the brain and the spinal cord. Okay, and we'll see that we have these different nerves that will branch off the spinal cord as well. So when we talk about different systems over the course of the upcoming semester, we'll see that features emphasized in particular. Okay, so when we talk about this peripheral nervous system, that is our yellow nerves coming out all the rest of the body. That's how we move stuff. That's how we feel stuff. All that kind of good jazz. All right, when we talk about that, we've already introduced the idea that we can categorize a lot of neurons by virtue of being sensory or motor neurons. So one of the key things here to identify is that in the peripheral nervous system, we particularly call these terms nerves. There's not a whole lot of nerves that we refer to in the central nervous system, so it's a nomenclature thing. By nomenclature, we mean naming. Okay? So we can have motor nerves and sensory nerves, just like we talked about last class, motor neurons and sensory neurons in the central nervous system. In this particular situation here, okay, we can call these nerves the bundles of axons of a bunch of different neurons, okay? And the idea, just like we talked about, motor neurons are taking information from the spinal cord and sending it out to whatever it is we need to move by motor activity. And vice versa, our sensory nerves are taking the information from our periphery, from our fingers, our toes, our elbows, our knees, pretty much everything, and taking that information and sending it into the spinal cord, where it rises up that proverbial elevator of the spinal cord and goes into the brain for our central nervous system processing. Okay? So we can also categorize some functional elements of the nervous system. So we've categorized two basic delineations or taxonomies, being the central versus the peripheral nervous system, but we can also categorize the somatic or the autonomic nervous system. So by somatic, we're basically referring to those nerves that are interconnecting the brain and the big muscles and sensory systems of the body. Okay, in the autonomic nervous system, we're basically referring to those nerves that are connecting to the viscera. So what we mean by viscera, that's kind of a crazy word, probably never hear it again in this class. We're basically referring to those internal organs. Okay, so we'll take a closer look at exactly what we're referring to with that in a minute. Now, within the somatic nervous system, we can see cranial nerves and spinal nerves. So as you might imagine, spinal nerves are those that are basically connecting to the spinal cord, and the cranial nerves are those that are innervating the head, sometimes the neck as well. Okay, so directly from the brain. These are just some basic terminologies, vocabularies, sort of orienting how we can organize our understanding here. Okay, now if we stick within the cranial nerves here, okay, we can divide those or delineate them based on some specific structural and functional differences. Okay, there's a handful of them. A lot of other classes, at least in my heyday, I guess my heyday might be gone now. I hope not. Hope I still got it, but in case I don't, back in my heyday, we used to have to memorize all the cranial nerves. You're not going to have to memorize them all here, but I'm going to introduce them to you so you have some familiarity with them. Okay? So we can categorize the cranial nerves in a couple different ways. One, we can categorize them based on basically what they do. So you can have an olfactory nerve that's involved in smelling. Okay, we can have an optic nerve that's involved in seeing things. Okay, or what we mean by the vestibular cochlear nerve. Okay, that's going to be involved in transmitting information that we use to hear things and keep our balance in three-dimensional space, sort of proprioception. But we can also categorize the cranial nerves by virtue of numbers. Okay, and we use Roman numerals to categorize them. So you'll hear people talk about the 5th cranial nerve, or the 7th, or the 10th, or whatnot, but they can interchange with the names. Okay, so the optic nerve versus the 2nd cranial nerve, same thing. Okay, this is a schematic of them. Is this your question?
One more second.
Yes. Although all this stuff here that's on these slides are generally in your text. Okay, so the idea here is that we can just illustrate them and kind of show you a little bit of a navigation around these cranial nerve systems. Like I said, you're not going to have to remember all of them rotely, but we will focus on in the class, particularly on a few of them. The vestibular cochlear nerve. We'll mention the abducens nerve. We'll mention the trigeminal when we talk about taste. We'll mention the optic nerve and some of the olfactory nerves. So be familiar with them. Know where they are. We can see this is basically what we're going to call a ventral view of the brain. So we're looking up from the bottom. So if I were to lay down on this table and my feet, the bottom of my feet were to face you guys, you would be looking up. Am I right this way? That's the view that we're looking at here. Okay? Now, we can see these different projections, these different connections. All these little lines represent those cranial nerves. Okay? If you look online, if you want to memorize them all just because you really just want to get your nerd on and get this stuff down 100% extra, okay, there's all sorts of fun abbreviations and little mnemonic devices that people have developed in med school and anatomy classes to remember all of these. You don't have to do them for the purposes of this class, but they're out there and they're kind of fun. You can check them out. Okay. So that's kind of the idea. You might have something like the trigeminal nerve here, okay? Might be innervating through here. Tri, meaning 3 branches, okay? Let's see here. The vestibular coccylar nerve, okay? Anybody who's not familiar with reading Roman numerals, V is 5, 1, 2, 3 is 8. So 3 plus 5 is 8 cranial nerve, okay? We'll see that's within the inside of the way inner ear. That's going to be what transmits information from our hearing and vestibular system in through that little nerve to the rest of the brain. Okay? Yes, ma'am.
Is it around the sensory nerves or the motor nerves?
I'm sorry?
Should we love the motor nerves to like where they are and what they do, or is it more sensory? Could you just point out the sensory one?
Yeah, you're not going to have to know like the names really of either like the motor or the sensory nerves, but it's important to know the information. You know what I'm saying? So yeah, you're right. Right now we're just talking about the sensory elements of it. Okay, so this just goes through and lists a handful of other ones, but they're all listed in the figures and the text in the book, so we're not going to belabor that here, but just to introduce that we have these different elements. They have weird names: abducens, hypoglossal, ocular motor, okay? Oculo meaning eye system, for the eyes, okay? Motor meaning you're going to move them. So when we move our eyes, we need something to do that. That's going to be the nerve involved in that. So we have this. This is basically just going through those. The vagus nerve, if any of you guys remember old Ivan Pavlov and his dogs, okay? He was stimulating the vagus nerve in that associative learning, classical conditioning. Okay, so we see some integration here across multiple classes, hopefully, that you guys are taking. Makes sense? All on board? All right, let's keep moving then. Okay, so we mentioned the facial nerves, or the cranial nerves, that is. We also have a bunch of spinal nerves, okay? 31 pairs of them. They're going to project from the spinal cord. You're going to have a group of sensory fibers that also enter the spinal cord, okay? So we can categorize them along different lengths of the spinal cord, okay? So we have different names for those sections, okay? If any of you guys have been to orthopedic doctors or had, chiropractors or whatever, you're just interested in the back, you'll see that there's different vertebrae that start with different letters that refer to the cervical, thoracic, lumbar, sacral, or coccygeal segments, okay? And we'll take a look at exactly what we're talking about with that, okay? So this is just an illustration here. The colors are really helpful. So you have the cervical section, okay, kind of the neck area. Okay, that's going to be this top area. So you have this is colored in red, and this is colored in red. We go a little bit further down, and we see the thoracic section, sort of that top part of the back. Okay, this section is colored in blue on this color scheme, and it's blue here. We move a little bit lower. Okay, you see the lumbar section. Okay, sort of where that inside curve of your back is. Okay. So if you're in a crowded bar, that's kind of where maybe people are walking past you where they're going to kind of tap you and move past you. Don't you hate when that happens? Okay, so it's going to be sort of on the lumbar area of the back. And then when you get further down by the tailbone, you get the sacral and coccygeal areas. Okay, now we can see that the spinal cord, of course we know it has that sort of S shape, a little bit, or J shape, depending on how you look at it. And we can see that when we slice that up, because the spinal cord is part of the central nervous system, we can see that it looks different ways at different layers of the rising and falling. By rising and falling, we mean ascending or descending from the head down to the tailbone. What do I mean by that? Let's take a look. Okay? See up here in the cervical area, you see a bunch of this sort of gray area in the middle, okay, where a lot of nuclei, neurons are. There's a lot of white matter on the sides. White matter represents the tracts of axons that are myelinated. Remember we talked about myelin? That fatty sheath? It's white. So it turns up as white matter. And the nuclei that are not myelinated, they're gray. So we have this classic delineation of what you might call white matter and gray matter. Pretty simple. Well, look here. See how that gray matter got real tiny? You have these different horns that kind of look like horns. Well, they call them horns, okay? And the white matter is enlarged. You have a lot more axon tracts packed into that little section, and you can see through these slices, see how it gets a lot bigger here, a lot more nuclei? It's going to kind of wax and wane as you come up and across. So there's sort of some fluid differences as you move ascending and descending through the spinal cord, okay? And what it helps you do is later on when you guys become professional neuroanatomists in your careers, okay, you'll be able to look at this slice or that slice and just look at the shape and the morphology of that slice on a screen or a microscope, and you'll know exactly what section of the brain, or excuse me, the spine that's from. You're not going to have to know that for the test, but just some orientation, some sort of geography or anatomy of what we'll be looking at. Okay? Now this is a closer look into the spinal cord. Okay? Now the spinal cord is really important. We all obviously, hopefully already can respect that fundamental fact, right? Because we all like our spinal cord. We don't like when it gets hurt, and we get scared when it gets hurt because it can risk paralysis because you disconnect a lot of these neurons. Okay? Because it's so important and an essential element of our nervous system, we have all these vertebrae, these bones that encapsulate it to help protect it. But because we're not robots and we can't walk stiff, or at least we shouldn't need to walk stiff, right? When we're surviving in the jungle evolutionarily, we need to be able to move around. There's all these breaks within the protective bone. So each vertebra or bone is separated by some cartilage that allows it to flex and do crazy breakdancing and everything else that's fun. Okay? So we can see the vertebra, basically the bone. And if you start breaking down this spinal cord, see the little horns up there that I pointed to in those earlier slides? Right? You can see there's gray matter on the inside and the white matter on the outside. This is a column, right? The spinal cord. It's a column going up and down, but it's encapsulated by several sheaths. We'll look at this later in the brain as well, but these sheaths are basically membranes, okay? The membranes are there to protect it, okay? And the membranes can be subdivided into 3 different kinds of membranes, and we'll see this. They also cover the brain too. They're called the meninges in general, and the meninges have 3 levels, okay? You basically have the pia matter, the arachnoid, and the dura matter, okay? They surround the nervous system and they protect it, okay? So we can see those layers being peeled back, all right? So when you peel them all back, we get to the actual spinal cord, initially protected by bone and many layers of membrane, okay? That spinal cord, okay, has many nerves coming in and out of it. We can also categorize another feature. If we walk back here, we see there's some words here, ventral and dorsal, okay? Ventral, based on this 3-dimensional rendering, means more forward or towards the face in this regard, or towards the chest. Dorsal is going to be on the back, more towards the back end. Easy way to remember that is a dorsal fin of a shark or a porpoise or whatever. All right? If any of you Ace Ventura fans are out there, there's a famous scene in the old classic movie where he talks about a dolphin having a dorsal fin. He says it in a crazy German accent, and it's funny. I'm not. But you can go see him do it. It's a lot better, more entertaining. Okay? So we have a dorsal. Do you have a dorsal fin? It's in the back. Okay? So we can see that there's a dorsal way and a ventral way that it connects. These nerves connect to the spinal cord. Okay? This is a much closer look. Okay? Now remember I said we have the gray matter in the middle. We have the white matter surrounding. Okay? And you have dorsal and ventral directions. And so you can use that direction information to categorize the features of what's happening with the spinal cord. In particular, we have what's called roots and ganglion. So the roots, just like you might think of for a tree or a plant, the roots, the ones on the dorsal side, surprise, surprise, are called dorsal roots. Anybody want to know what we call the roots that are on the ventral side? Ah, yes. Awesome. You studied, didn't you? You do it. Who'd have thunk it? Okay. So, dorsal roots, ventral roots. Okay. We also have these big blurbs, okay? They're collections of nuclei. Call them ganglions. Yes, ma'am?
When people have like nerve damage on their spine, what is it that's different between theirs and that image or the other image?
When people have nerve damage in their spine, what is it that's different between
Like
this?
when they're paralyzed and stuff, where they can walk with functions. What is it that's damaged or different?
Well, at the simplest level, so lots of different people can have lots of different types of damage, which makes the question really, really difficult to answer in a comprehensive way. But one way you can think about is it could just be cut, partially or in full, that doesn't allow the axons that are covered in white rising up here to connect into the brain and tell you what you're feeling. Or that doesn't allow the axons that are descending and coming back down to tell your arms and legs to move. So at a very simple level, that severance, or cut, is one way that you can see those injuries manifested. Okay? So we have this basic style of organization. Okay? Now, we can also look at that other system that we talked about. Okay? The autonomic system. And even that has two further subdivisions. Okay? We have the sympathetic nervous system and the parasympathetic nervous system. Now, we can look at that in a couple ways too. Okay? Now, the autonomic nervous system is involved in controlling the body's organs. Okay? Now, that division has different systems as well. Okay? So our sympathetic nervous system is innervating what we call the sympathetic ganglia, those clusters outside the central nervous system. And the way that you can remember it, its key feature is to be involved in controlling our fight or flight response. Okay? So the sympathetic nervous system is involved for a sort of reaction to danger. Right? Something happens and we need to know, raise the alert of the body system, prepared to run away or throw down. Because something's about to go on right now. You got to decide if you're going to run, or if you're going to stand your ground and get it on. Right? And if you're going either way, you got to get your stuff together, right? That's what's happening here with the sympathetic nervous system. Now we can contrast that to the parasympathetic nervous system, which is kind of like after all that has happened, we need to restore things back to kind of like chillaxing a little bit. Okay? And it's more involved in sort of recuperation and relaxing those elements that had been activated by the sympathetic nervous system. Okay? So we can take a look at how they affect and connect to different different elements of the body, as well as how they can operate through different neurotransmitter systems. Okay? So this is kind of what we're referring to. Okay? Now, you don't have to know all these organs and all that, but they're here to show you the stomach, the liver, the heart, lungs, what have you. You've got the sympathetic nervous system on this side and the parasympathetic nervous system on this side. Okay? Now, they're going to operate through different neurotransmitter systems. Okay? So, basically, cholinergic neurons, what we're referring to is a neurotransmitter called acetylcholine. So acetylcholine, cholinergic neurons that use that chemical to communicate, versus what we'll call noradrenergic neurons, which are neurons that use the chemical called norepinephrine. Okay? So a lot of thick vocabulary there, but the idea there is that they're either stimulating or relaxing different features of the body. Okay, so let's just say that if sympathetic nervous system, you might have the pupils dilate, okay? Or you might have the pupils constricted under the parasympathetic nervous system activation, okay? So, fight or flight, your pupils might constrict, okay? Or, excuse me, open, dilate, because you got to see what's happening. Oh, crap, what's going on? Right? That's why you get like the cartoon image of like the eyes bugging out, right? When the cartoon characters are scared. Oh crap, what's happening? Okay. Basic idea there. We can also see, look at some of the morphology of the shapes here. We have the sympathetic nervous system basically innervating these systems by virtue of these middle levels of the spinal cord and the nerves. Okay? The parasympathetic system are operating a lot through the cranial nerve system, as well as down here in the sacral and coccygeal nerves. Okay, just some organization here. All right, know that basically this delineation, which one's involved in activating, which one's involved in relaxing, and some of the chemicals involved in transmitting them. Okay. Okay, so let's move into the brain. Okay. We will talk a lot about some brain anatomy. I'm going to do so with the assumption that you've already taken a look at that through the assignments and the homework provided on the syllabus. Okay? So we have one big way that we can categorize different features of the brain, the left side and the right side. So what we can observe just by looking is that there seems to be two halves. We'll call them hemispheres. And we can look at those hemispheres just from the outside and see that they're basically representing what we're going to call cortex. Okay, so you can have a cerebral cortex on the left and a cerebral cortex on the right. Okay, now those cortical areas can be further identified by just looking a little bit deeper and seeing that there are some special features that seem to become evident. Okay, the cortex isn't a bunch of flat tissue. It's a bunch of convoluted, folded up, weird stuff. Basically, ridges and valleys. We'll take a close look at that. We're going to call them gyri and sulci. Okay, so the gyri are the raised parts, and the basically sulci are those valleys. Okay, now we can categorize those hemispheres in further divisions. Not just left and right, but you can also categorize that there's a frontal lobe. There's also basically some lobes that are posterior to the frontal lobes, and we'll talk about their division. There's a parietal lobe kind of on top, an occipital lobe in the back, and then a lobe on the sides called the temporal lobe. We'll look at some figures for that too. Okay? Okay. And we can categorize some basic boundaries, some landmarks that we'll point out too. The Sylvian fissure and the central sulcus. Okay? Fissure basically is referring to a sulcus. Okay? It's kind of like a big line. Like, you know how we're here on the San Andreas Fault? That's basically a fissure. Okay? It's a big sulcus in the geography world. Okay, so let's take a look at some of these classic hallmark features that you should be familiar with, be able to identify, know where they are, and know where you would find them if somebody were to refer to them in discussion. Okay? So here we have basically the left cerebral cortex. Okay? And we can see those four lobes divided. They're important four lobes to know. Frontal, parietal, temporal, and occipital. The frontal lobe, surprise, surprise, is up here in the front. I feel like Vanna White in Wheel of Fortune. Okay? And if we look to basically the bottom and the side, this is the temporal lobe. As Vanna White strolls across to the other part of the Wheel of Fortune, we can see all the way in the back is the occipital lobe. We'll learn later that it's heavily involved in processing vision. And up on top of that, in between the frontal and occipital lobes, are the parietal lobes. Okay? One on each hemisphere. So there are four big delineations of the cerebral cortex. Okay? Now, we talked about those big fissures. Okay? This is a Sylvian fissure. It cuts all the way across. And we'll look in later how it actually can be sort of pulled and divided. It's not an arbitrary fissure. It's a huge sulcus. The gyrus are these folds. This is a gyrus. This is a sulcus. The black lines are the sulcus. Easy way to remember that, right, is when you're doing really well, you're pretty happy, right? But when you're kind of depressed and you're down in the dumps, you're sulking. So you're down in the dumps and you're sulking. Okay? So you're down in that valley in the fold. Okay? When you're up on top, you're on the gyrus. Anyways, okay, we also talked about the central sulcus as well. So remember we talked about that here? The central sulcus dividing the frontal lobe from the parietal. That's what this dashed line is here. Okay? So it's a huge sulcus that divides this section from that section of the cerebral cortex. And we'll see it's a really important functional division too, which means not just in structure, that it's separating the pieces of the brain, but the stuff on each side of that huge sulcus is very, very different in how it processes the function or the operation of the brain. Basically, this is going to control some movement, and this is going to control some sensation, okay, or process sensation, or process movement. Okay. Now, because this is before the sulcus, and this is a gyrus, we call it the precentral gyrus. Likewise, this gyrus that's after the sulcus is the postcentral sulcus, like a post-game show, after the basketball game, after the football game, post-game show. Post, pre, relative to that central sulcus. Now, a couple other big features that we'll dive into, okay? This is not all there is in the brain. There's actually a ton of stuff underneath this top layer of cortex, but we'll get to that in a little bit. We also have this big chunk of tissue here. It's called the cerebellum, okay? It means little brain, because it's an additional section of nervous tissue, and it's also part of the brain as well. There are more neurons in here than there are in all of here. And 95% of all studies of the brain focus on this, historically, not this. Pretty crazy, huh? So if you want to go off and plant your flag and discover a bunch of new stuff that not a lot of people have known, this is an area ripe for discovery. There are a lot of people working on it, but not nearly as many that are working on everything in here. And there's a lot that we don't know yet about this. It's involved in lots of things that we have not characterized very well. That's a point of excitement. So that's a challenge to you guys to go figure that out in your careers. I think that'd be pretty cool. Let me know what you find. Okay? It's heavily involved in control of motion and balance and a lot of other things in cognition too. Also, the brain is not operating in isolation. Stuff has to come into the brain and stuff has to come out of the brain in terms of signals, right? We talked about this connection to the spinal cord. It connects to the spinal cord by virtue of the brain stem. Okay? You got this little bump down here called the pons. We'll look into that in more detail. But this is the brain stem, and so we'll see evolutionarily, the cortex is the newest part of the brain in evolution over millions of years. These areas down here are the evolutionarily older sections of the brain. Okay? Heavily involved in the most simple of functions. Breathing, respiration, controlling heart rate. Okay? So when you get tumors and lesions in these deeper, older parts of the brain, they can be very, very dangerous and fatal, because if they're pressing on this older evolutionary area that's involved in respiration, breathing, and it ruins those cells or makes them dysfunction, you can't breathe, you're out. Game over. You get a lesion up here, like Phineas Gage, things might change for you a little bit, but you're not dead. Right? Okay, so again, different structures and functional relationships. Okay, we talked about these different gyri and sulci, the postcentral gyrus and the precentral gyrus. There's also a huge chunk of axons that connect both hemispheres. So you have the right hemisphere and the left hemisphere. They're connected by a huge chunk of connections of myelinated axons that we call the corpus callosum. Okay? And that's what allows the two hemispheres to talk to each other in large part. Okay? And communicate across left and right sides. Okay? Now, a couple important terms to mention as we orient to our movement through the nervous system. Some of this stuff we've already introduced a little bit in some of our earlier discussions in our earlier slides. Okay? Some of them are going to be new, and we'll visit them as well. Do I need to slow down at all? We're good? Okay. Yes, ma'am. Back one more. Is it? Yeah, this is it. Yes.
Okay, so the precentral gyrus?
Gyrus. Yeah.
And the post one for, like it says on there, like what they're for?
Well, it's not, I don't know that I would say what they're for as the best way to talk about them. We can identify things that they do, and we can identify the place where they are.
Oh.
Does that help a little bit?
Is that kind of like what matters to know?
Yeah, yeah. So, again, this is our precentral gyrus. This is our postcentral gyrus. They're big landmarks. That's another way to think about them. What we're talking about right now are the big landmarks. So if we're going to divide the United States between left and right, what's our landmark? God bless you. What?
Left and right?
Yeah.
Mississippi.
Yeah, the old Mississippi River, right? That would be kind of like a sulcus here, right? That's a pretty big landmark between the left and the right sections here, between the frontal lobe and the parietal lobe. Really big sort of slice across it, right? Now, I guess somebody could have also said the Rocky Mountains would have been more like a gyrus, right? But it's not quite down the middle as much. But the idea, you guys see the idea there? Okay, so let's move on and talk about the ways that we'll refer to how we look at things. Because as we go through the rest of the semester, or quarter, I call quarter semester, sorry guys. We'll talk about lots of different ways that we can look at the nervous system, because some ways are really, really good to look at different features of the brain, or different parts of the brain, when we talk about motor control and movement. But that same way to look at the brain might be horrible and totally not as useful when we talk about memory, and vice versa. So we got a little every angle, right? Okay, let's start off. Okay, so there's three main ways that we can slice up the brain that we can show how it looks: sagittal, coronal, and horizontal. Okay, so sagittal means that we're cutting the brain into left and right halves. Okay, so if I'm standing this way, and you take a buzz saw after your test and you hate me and you're like, buzz saw this guy right down the middle here, open me up like that. Okay? That's kind of looking like that view. Sagittal. Okay? We can also look at a coronal plane, which means cut me this way, and have the front part of me here come down like a trap door and look at me this way. God bless you. This is coronal. You might see it shaped like that. We'll look at some ventricles that might fit in there, and the spinal cord will be coming up this way, and you're going to have the cortex on the outside here. Okay? It's going to be coronal. So this would be sagittal. This would be coronal. We also have a horizontal plane. Okay, so that's basically cutting it between this way and then popping my head up this way like a Pez dispenser. Okay? So you can look sort of down or up, depending on how you prefer it. And you might see just a generic way looking this way. Okay, so you're looking down. Okay, now my drawings are terrible. I'm not going to subject you to have to suffering through them. We'll show some basic pictures that are a lot better ways to look at. Okay, now we can also talk about different orientation terms. Medial means towards the middle. Okay, this is the middle. Okay. The more towards the middle it is, or the more towards the midline, the more medial a direction is. If you move towards the sides by the temporal lobes over here, that's going to be lateral. Okay? Now, we can also refer to things like ipsilateral and contralateral. Contra means opposite. So it's the other side. Okay, so my left arm is typically moved as a function of neural activity happening on the contralateral side, or the right hemisphere of the brain. But it also gets some contributions from the ipsilateral side, which is the same left side of the brain. Make sense? Contra means opposite side, opposite hemisphere. Ipsilateral means same. Okay? Superior and inferior.
Okay? Superior, kind of closer to top. Inferior, kind of closer to bottom. Okay? Now, you can also look at the sides that's here. Anterior, kind of frontal, or front. And posterior, or more towards the back. Make sense? Okay, so you might talk about the anterior prefrontal cortex, the very frontal part of it. Okay, posterior parietal cortex. Okay, so you have your parietal cortex maybe here, but the posterior section of it back there. Okay, so there are ways to tell your friends what your favorite part of the brain is. And you're like, yo, what's your favorite part? I like the posterior parietal cortex. Really? I like the inferior prefrontal cortex. Oh, really? Yeah, that's what I think. Well, I tend to think that the superior temporal lobe is a pretty awesome area instead. So it's a way to tell people where are you talking about when you're referring to some feature or function or element like that. Okay? Guiding people to where they need to go. Now, there's also other terms that you might see too. Rostral or caudal can typically be interchanged with anterior and posterior respectively. And it's typically derived from the animal literature that a lot of anatomy is taken from too. When we look at rodent brains or monkey brains, you might refer to the caudal or the tail side or the rostral or the head side. Now, we don't have so many tails when we look at humans because we've kind of lost that feature in evolution. So we tend to talk about anterior and posterior more. Yes, ma'am. The third one down. How do we pronounce that? Huh?
Ipsilateral.
Ipsilateral. Yes. So lateral means side. So any football fans in the room, you'll know that if you watch the Super Bowl, every now and then, maybe you might see a lateral throw, which is different than a forward pass. Why? Because a lateral is pitching it off to the side. To the side, lateral. Ipsi versus contra. I find it easier to remember what contra means. To know that contra means opposite. Therefore, I don't have to remember what ipsi means. I just have to know that it's not opposite, which means same. So that's my convoluted way of, I have to remember it. But you could just know that, hey, ipsilateral means the same. Make sense? Okay. So we can also look at other elements as well. Proximal, especially when we're talking about the peripheral system. All right? We talked about our spinal cord here, right? Remember we had a slice of the spinal cord in our earlier figures? Okay? Now you have the midline across the middle of that too. And you can have something be medial or lateral, just like we have medial or lateral to the brain. But we can also categorize it or refer to it in the context of being proximal or distal. Okay? Very similar to medial lateral, but in this case, more towards the spinal cord sort of conversation. Okay? So proximal or distal. Proximal means more towards the center. Distal means more sort of laterally. Okay? Or towards the periphery, further away. We can also look, as we've already introduced, to these terms dorsal and ventral. Okay? Dorsal is going to be more towards the back. Do you have a dorsal fin? Like Ace Ventura said. Okay? Do you guys know Ace Ventura? Is that too old of a reference?
I know.
Okay. We got one. Okay. You can tell everybody else about it later. Okay. So it's a funny show. Old funny. And ventral meaning towards the belly side or the front. Okay? Yes, ma'am.
When we talk about the brain, though, would dorsal and ventral be interchangeable with superior and inferior?
It is, in fact. So Rose asked, when you're talking about the brain, does dorsal and ventral interchange a little bit
with superior and inferior? And the answer is yes, and that's tricky to think about. However, it's because of this sort of change that happened in evolution. Let's say when we talk about animals, we talk about dorsal and ventral in that particular context, because a lot of times they weren't walking upright on 2 feet, and their nervous system went straight across, right? Imagine your dog or your alligator walking on 4 feet, right? So the spinal cord is going this way, and the brain is also going this way, and it's generally the same. But in us, our brain is kind of still oriented this way, but the spinal cord changed. Make sense? We'll take a look at that too. Okay? And we'll see dorsal and ventral pathways when we talk about vision and things like that, and attention. Okay, so here's a lot nicer ways that I promise you won't have to pay attention to all my crappy, horrible stick drawings. Okay? So, this is what we have. Everything I just talked about. If you slice it this way, or this way. However, there's basically 3 ways to take a buzzsaw through my head. Okay? This way, or this way. Okay? Sagittal, cut me this way, open it up, see the whole anterior to posterior side of the brain. Coronal, it's going to be over here. You would look in here. See how this kind of looks like that? Yeah? And then this horizontal plane, cut this way, was what was drawn over here. And of course, we have our sagittal view. Yes, ma'am.
Is this picture in the book?
Yes, ma'am. Okay. In fact, it's box 2.2 in the book.
Oh.
Page 35.
35?
35.
35. Okay. We can also, we've already introduced the idea of gray matter and white matter, so I'll skip over this because we've already talked about it. And this is a close-in view of white matter and gray matter differentiated now in the brain instead of the spinal cord. So we used to delineate white and gray matter here in the spinal cord. Remember, we had our gray matter, the nuclei, those cell bodies kind of in the middle here. Okay? But we can see that same element here. Cell bodies all along the top of the cortex, sending their connections across to the other hemisphere by the corpus callosum. And these are all the connections from all these areas. The white matter myelinated, connecting everywhere else. So you can see this is a deep, dark area where there's a lot of cell bodies. We're going to call it the basal ganglia. Okay, we'll look at that later. And there's strips of white matter that go across and connect. Okay? Yes?
So the axons are probably a lot longer than the pictures would lead you to believe, right? So I get
Yes.
I get a little confused when I think about it, because I think of the gray matter. If it's just the cell bodies, I imagine it would be really thin, the gray matter layer. But I'm imagining
Well, it's relatively thin. It's this thin layer on top relative to this huge thickness of the brain. It's all relative, right?
Yeah.
But you're right, right? Sometimes we can misrepresent in our head the way that the reality of it is. But what you're right about is that the gray matter, the nuclei are very, very creamy. So it might be a really thin layer. However, there are billions of these tiny things and they're packed and stacked. Okay? Billions of them, and they're packed and stacked on top of each other. So even though they might be tiny microscopic cell bodies, there's so many of them that they stack up and form a layer. And you're right, some of these are long distance axons, but we'll also see examples of sometimes when they're short distance and medium distance and all that kind of jazz. Okay. So, let's talk a little bit more about these structures now. Back here, I referred to things like the basal ganglia and how there's all these different elements in the brain, and they develop in different ways, sort of evolutionarily, but also in terms of development too. Okay, so we have a lot of crazy terms that are referred to in development, and particularly calling different areas of the brain different things. Okay, so when we start in development, okay, we start talking about basic elements of the nervous system that begin to develop at the tiniest level of the embryo. Okay, we start to see what becomes the spinal cord in the brain initially starts off as just a little tube of stuff. Okay? We call it the neural tube. Because it seemed to make sense when we needed to name it. What are we going to call this tube of neural tissue? I don't know, Bob. Why don't we call it the neural tube? You know, that's a great idea, Steve. Let's do that. Okay? Which is really helpful because we already got our hands full of a bunch of crazy terms to remember. Right? So, the initial little tube in development that becomes the nervous system is the neural tube. It can be divided into sections because it's a tube. Okay? There's a part of the tube that's all the way in the front. We'll call it the forebrain. There's a part of the tube that's all the way in the back. We'll call it the hindbrain. Right? And of course, there's a section of the tube that's in the middle. Okay? That becomes the midbrain. Okay? Now the forebrain, that chunk in the middle, ends up having several other little divisions within it too. Okay? And the two main ones we'll refer to are the telencephalon and the diencephalon. Okay? The hindbrain also gets subdivided into other areas. Okay? So, if we think of our tube, right? And let's say we're going to call this the forebrain part of it. This is the hindbrain part of it. So back, front, and of course middle. Everybody on board? Pretty simple, right? Our forebrain can have other little chunks within it, okay? We're going to call it the diencephalon. Hold on. We're just going to put an abbreviation there. And the telencephalon, okay? Now, the hindbrain can also be divided into 3 sections too, okay? We're going to have basically the pons, okay? The medulla, and cerebellum. Remember that little brain that has more neurons than the whole cerebrum? Cerebrum is different than cerebellum. These words are going to be confusing and all that, but once you just get used to it, it'll start to play out and make a little bit of sense. Okay? Now, when we talk about the brainstem, we're referring to these areas combined. Okay, the hindbrain, brainstem, we're talking about a lot of that, but together like that. Okay, so this is basically the brainstem. Because we already saw the picture that the cerebellum isn't really part of that stem. Right? Okay, so let's take a better look at what is happening. Okay? So I like to basically set expectations really low with my crappy, horrible stick drawings and get you craving and starved for something much better and simpler. Okay? Because if we can understand it a little bit with simple black and white stuff, then the colors just help us now. They don't become more confusing when we look at it. They become a relief that we don't have to look at this old guy's horrible art. Okay? So, let's look at the better guys, much better cartoons. Remember we talked about that neural tube developing over time? Here's that tube. It's just the tube is kind of like crunched over, because in development, the baby's kind of crunched over and, you know, there's not a lot of space for it to go in the belly, right? Okay, so kind of crunched over. You have that neural tube, and you got the front, middle, and back. Fore, mid, hind. Simple enough. This is over time. 25 days. Which is what? Just about 3 weeks. So it's pretty cool. It's really developing really, really quick. And 35 days, 40, 50, 100 days, okay, you start to see these processes happen. Now, as we're getting more complex, and as it's developing more, we're going to see more tissue and cells grow, right? Because we want to grow to be more than an embryo, right? We want to be all of us, right? And we need more than just a few cells in an embryo to be able to do our homework and drive cars and all that good stuff. So as this more tissue grows, there's not a lot of spot for it to grow because babies' heads are tiny, right? Thank God, right, ladies? Okay. So it's got to start getting crunched up and packed in and folded in order to save space. So it starts to fold in. This tube starts to fold. And the fold, as it starts folding in, right, this starts coming in here, and this forebrain starts coming in and wrapping up above it. Which is why we start to see that shape that we'll see for the rest of the semester. Make sense? Okay, so it's wrapping up above, and we can see these colors starting to refer to these different areas, which is the same thing that I drew with dotted lines. Yikes. Let me grab my marker. Alright. Does this help to sort of conceptualize what we're referring to? Okay, so we got the neural tube, fore, mid, hindbrain. That forebrain, midbrain, and hindbrain starts to develop at the same time as it's connecting to this yellow part we've added now to the picture, the spinal cord. Alright? Because the brain is not the spine. Somewhere it has to differ. That's just where that happens. As we get further along, 50 days, we start seeing that telencephalon develop in the forebrain, and this bottom end of it sort of underneath it, the diencephalon. We'll see later is the thalamus. Okay? So all this is showing is just colors in pictures of the bubbles of the areas that we talked about here. Okay, so don't let it, I don't want it to be like, hopefully not too confusing or freaky out. I'm trying to actually dial this in and break it down a little bit. Okay? And this ends up being much more closer to what we talked about. And so this telencephalon, diencephalon, we see that becomes kind of the cerebral hemisphere. Frontal, parietal, occipital, temporal lobe. Kind of looks like a football helmet because the football helmet is kind of designed to cover and protect these four lobes of the brain. Right? You can kind of imagine like a face mask coming across here. Yeah? Okay. So let's take some closer look. As it develops into a full-grown brain, these colors help to delineate it. Yes, ma'am?
On the other one, the side, is that from young to adult, or?
No, this is all prenatal. This is all in the belly.
Oh,
100 days.
okay.
Yeah, so that's what we got here. All the way over here. This is like 3 weeks in.
Because that's when.
Yes. Well,
Yeah, I know.
this person isn't pregnant.
No, I know. That's the.
Yes.
Okay.
Yes. Okay, so when we look at this, it's the same information. So I always got super confused by this, and I still get really confused by it. It's a really helpful way to actually simplify things, but I like to walk into this having this conceptualization, because it's the same information. It's this breakdown and the structure that we just walked through conceptually, just shown on the adult matured brain. Okay? So we've got the peripheral nervous system and the central nervous system. Where's the peripheral nervous system on this slide?
It's not.
Nowhere. It is nowhere. Right? All we're showing is one part of the central nervous system. I guess the whole central nervous system, in the sense that we have the token spinal cord back here. But really we're looking at the top part of the central nervous system, the brain. So we start from here, even though this tells us of that delineation that we already talked about, the somatic and the autonomic nervous system of the periphery. And the autonomic is divided between sympathetic and parasympathetic, acetylcholine, norepinephrine. Okay? We come up into the central nervous system and we break it down systematically. First, we've just got the brain. But of course, we care about more than the brain. We got the forebrain, the midbrain, and the hindbrain. From the neural tube, forebrain, midbrain, hindbrain. That's all we're showing here. Fore, mid, hind. The forebrain, remember we said, had the telencephalon and the diencephalon. The midbrain is basically just the midbrain. Remember back here, we broke the hindbrain down to the pons medulla cerebellum. pons, or pons medulla cerebellum, that's our hindbrain. Now we broke that telencephalon and diencephalon. Remember I referred to that as the thalamus. We'll also talk about the hypothalamus too. And then our telencephalon, we get that cortex, the basal ganglia I referred to, limbic system we'll talk about. A lot of crazy words. Let's see if we can break it down to some pictures. Is that preferable? Okay, let's do that. And these colors are these colors. It's really, really helpful. When you're ready to pick up your book and slam it on the ground and be like, I'm done with this, I'm so confused, I've had enough at some points during your studying, right? Pick the book back up, give it a little hug, snuggle up against it, be like, I didn't mean it, baby, I really didn't. Let's make up. I appreciate you, because you can appreciate that they actually break this down with colors to help us simplify it and understand. Make sense? Please don't throw your books down. Okay? Because hopefully what we're walking through will help to keep it less frustrating. You agree? I hope so. Okay. If not, talk to me and we'll help simplify it more. Okay. So we already talked about this idea that you can have nuclei and tracts. Okay? Nuclei are basically collections of neurons, and tracts are those collections of axons. Okay, so we're sort of revisiting this idea. But when we're in the peripheral nervous system, that is outside the central nervous system, we call that ganglia and nerves. Okay? There are exceptions to that, and it is confusing. Like the basal ganglia, you guys should have just raised your hand and be like, call BS on me and be like, no, you just called this collection of neurons in the brain, the basal ganglia. That's not the same as this. Ganglia is for peripheral. It's an exception. Lots of neuroanatomists and people have worked on this for hundreds of years in all sorts of different parts of the world. They worked on it in their own little silos. They didn't always talk to each other. Different names came up, and there are exceptions to the rules, just like anything else. Okay? When we talk about the cortex, it's made up of a lot of neuron bodies, these nuclei, and one of the key features of them are their pyramidal shape. Okay? And we already talked about that last class in morphology of cells, right? So neurons are shaped, at least in the cortex, oftentimes as pyramidal shells. Okay? They're the biggest and most prominent neurons in the cerebral cortex, and they send their axons not just to each other in the cortex, but across to other cortical areas through the myelinated axons that make up our white matter. Okay? Now, the key thing to add to the story now is when we're talking about our cortex, right? So this is our layer of cortex in our coronal slice. Yeah? Let's just look at let's take a strip of it. Let's grab a little element of it here. This little column, we'll call it, are actually divided up into several different organized features. All right? There's basically a columnar organization, but there's also basically layers. Okay? So you can see that there's layers within it. Okay? And so for this reason, one easy way to remember this is that, first of all, brains, but in particular the cortex, as well as brains, are like ogres. What are ogres like? Onions. Why are ogres like onions? Because they have layers, donkey. Okay? We're going to see that principle and property exhibited in several different capacities of the neuroanatomy. Both in gross neuroanatomy, there's layers within the brain, there's layers within the cerebrum, and there's also layers within the cortex. So there's layers of stuff like this, but there's also layers within the cortex. Okay? And we're just going to break it down. There's also these columns that we've talked about as well. We'll get into that a little bit later too. Okay. Here's an example of both columns and layers. Okay, because the cortex is like ogres, and ogres are like onions, and onions have layers. Layers. Now, because scientists like to get paid for doing something, they can't just look at the layers. They got to give them a name. They got to tell their boss, look, what would you do today? Well, I looked at some layers. All right, great. What about it? Well, what'd you do to get paid? Well, I gave it some numbers. Right? We got to show we do something. So you named it. And it's also really helpful to have names, right? So you number them. 1, 2, 3, 4, 5, 6. And then we go down to the white matter. Okay? Notice these thicknesses. Okay? There's different sections. There's an organized. It's not just a random haphazard thing. Although what we will see is it will begin to look random and haphazard in this systematic organization when we look at cases of neuropathology, diseases, okay, impairments. And if we look at some of those layers, this is one of those pyramidal neurons, and we can see that there's a shape. It tends to project up, but there's also lateral connections. Okay? And this shows these different layers of the different neurons within them. Pretty cool, right? Okay. So, that's our little schematic of our top layer down to the white matter. Now, there's also sublayers within there. There could be 3A, 3B, or 4A, 4B. Leave that for later. Yes, ma'am.
Is this what the gyros are made of then? Gyri made of?
Yeah.
Yeah?
So this is just this.
The whole
So-
thing though?
Yeah, so this let's see. So this hasn't been drawn. We have let's say this would be your sulcus, right? And this would be the gyrus.
Like if you were to poke the brain, would you be poking layer 1?
The top layer, yes.
Yeah, okay.
If you poked it from the top,
Yeah.
if you poked it from the bottom,
You'd be poking the white matter.
well,
Kind of.
potentially, yeah.
Okay.
And so, yeah,
Depending on
so
where you cut it, right?
exactly. So that's what this is showing. This is that top layer here of 1 all the way from the top. And this is coming down. So this is white matter. So this would be like towards the bottom here. That's why I did this
Okay.
schematic.
Okay.
Yes.
What's the difference between these and the meninges?
Well, the meninges are not neurons. The meninges are not part of the brain. They're the layers that surround the brain.
Okay, thank you.
They're like sort of like layers of not skin, but sheets. Yes.
Is the myelination inside those layers? Or no? Or is it something different?
Good question. The myelination is of the axons, particularly the ones that project down through here in the white body.
Oh, okay.
Yeah.
Okay.
Yes.
So all of those cell bodies have long axons that go down into the white matter.
Not all.
Most.
Some. Some have axons that just connect. So this is exactly what this is showing. Some just have axons that connect to the next layer. The next one. Local processing. Remember we talked about local versus distributed processing? This is why we went through that whole back and forth story of the battle royale in the history of neuroscience. Local and distributed processing. You could have axons being sent to local connections, and then axons being sent to sort of distributed, totally different areas that are going to have to travel with white matter. So you can have local processing within this layer, and then another type, they might have, processing back and forth within it, but then you might have distributed that goes across the corpus callosum to talk to this side over there. All of the above.
So there's no myelin now in this part, right?
Typically not, because it typically doesn't need it. Myelin is used to speed communication when the electrical activity would drop down.
Oh, okay. That makes a lot of
Yeah.
sense. Got it.
Yeah, great question. Okay, so let's look at some of these other structures that are promised, because this whole coronal way is a nice way to think about this topic. But we're going to start looking at the layers, not within the cortex, but now within the brain, more structurally. Okay, because the brain is like ogres, and ogres are like onions. They have layers, donkey. Okay? Yeah, sure. I'm not just weird randomly. Usually it's because I'm throwing out a movie reference or something. Okay? I am a little weird normally, but there's often a method to the madness. So, let's talk about some structures. Okay, we talked about the basal ganglia earlier. Remember we kind of showed that it was sort of this area, and there was a bilateral thing to it? Okay? This is going to starve you to desire some much better pictures that are on the way. Okay? So, the basal ganglia is composed of several subcomponents. So ganglia just means a collection of nuclei based on what we observe. And so it actually represents, the term basal ganglia represents a network of a bunch of different nuclei. Okay, we have different names for them. Part of it is the caudate nucleus, another part of it is the putamen, and another part is the globus pallidus. Okay, now it consists mainly, it does not say consists exclusively, it says mainly. There's other parts that are considered by anatomists to include the basal ganglia. Parts of the substantia nigra, subthalamic nuclei, and some other smaller network areas. Now we'll also talk about something called the limbic system. Limbic system is an old sort of Latin word that means around the rim, because there is this C shape, right? It's kind of like a C, like a backwards C to the brain, okay? And it's kind of considered the rim, okay? And these areas involved with the limbic system tend to oftentimes be involved in supporting a lot of emotion and learning and memory, but not exclusively, just oftentimes heavy players in those processes. Some of the areas in the limbic system that we'll talk about are the amygdala, the hippocampus, the cingulate, and the olfactory bulb. Okay? Now we'll look at pictures of these, okay? And we'll see that basically the hippocampus, this is kind of a good representation. So the hippocampus might be sort of this, a structure that wraps around like this, and at the very anterior front of it is a little almond-sized structure called the amygdala. Again, heavily involved in emotion, fear processing, okay, perceiving smells, okay. The hippocampus and the fornix that's associated with it, involved in a lot of learning and memory. Now, there's cortex up here that wraps around, called the cingulate, that's heavily involved in executive control, sort of mental discipline, attention. And the olfactory bulb, we'll see, is involved in smell. Now, this is a much nicer way to conceptualize it than my chicken scratch. Okay? Here's what we're looking at. First, we're looking at the basal ganglia. Okay? So these are deeper layers, Donkey. Okay? We're not talking about the cortex up here at the top. We're talking about these deeper layers now in here. Okay? Not the cortex at the surface, or the superficial layer. Yes, ma'am? No, just deeper. Yeah. Well, so you could think of it sort of more core-like, but I would just say deeper structures in the brain, as opposed, right? So, like this is the superficial or top layer. These are deeper. This is what we're looking at. This is the basal ganglia shown in blue, and there's several parts of it. Okay, we have the caudate and the putamen, the globus pallidus. Okay, the globus pallidus is sort of this and this, and there's an internal and an external version of it, or what they named it. There's an internus and externus. It's a little bit easier to see in a coronal slice that we'll see later, which is why we have these different planes of viewing the brain. I'll get the question in just a sec. The putamen is sort of this larger area, and then you have the caudate, which is sort of wraps around. You have the tail of the caudate. Okay? Now, there's also part of the diencephalon that we referred to earlier called the thalamus. The thalamus is what's shown in red here. Okay? Now, I should say, remember that there are two hemispheres in the brain. Everything we're talking about here, there's a left and a right side. There's a left one and a right one. There's a left thalamus and a right thalamus. Now the thalamus is going to become one of your near and dear friends, because it's going to be involved in all of our future conversations about different sensory systems. one of the things the thalamus does is it's basically the relay station. Whenever information comes in from our sensory peripheral nerves up the spinal cord, and it wants to go to a cortical area to do something and tell us information, it can't go there until it first stops at the thalamus and pays a toll. Thalamus says, bring it all here first. I'm going to then serve kind of like a relay station or relay hub and redirect you to the right part of the cortex to process a particular sensory piece of information. Oh, is it visual? Oh, then I need to redirect you to the occipital cortex. Oh, this is auditory information coming in. I'm going to send you the temporal lobe. So the thalamus is really important for that. That's this red sort of bubble. There's this subthalamic nucleus, this little green sucker I talked about there. That's also considered part of the basal ganglia. And we can see the fornix sort of wrapping around, or well, not so much in this picture actually, but this is that amygdala that's going to come around from the limbic system, that little almond-sized structure that I pointed to there. All right. And I did mention the substantia nigra earlier too. It's in here. That's in the midbrain. Okay. Substantia nigra is a crazy word. Just means black thing. Anatomists we're looking at on the slide. It's this area of cells that just stain very dark colored. So what are you gonna call it? I don't know. Area of black looking cells. Okay. Cool. So sort of our overview in this case of the basal ganglia. Now here's that limbic system. Okay? You can see the amygdala again. There's the amygdala here. Here's the amygdala here. Just like I drew it over here at the anterior part of this wrapping around C structure of the limbic system and the hippocampus. Here's the hippocampus. It's green. Okay? Hippocampus is one of my favorite areas of the brain. It's involved in learning and memory. I study it very closely. It's near and dear to my heart. And it's etymologically derived from the Greek word for seahorse. Okay? They thought it looked like a seahorse, and I'll show you some pictures of that later as we move along. Okay? Here's that cingulate gyrus. All right? The cingulate gyrus can be further subdivided between an anterior cingulate, a posterior cingulate, and a middle cingulate. Okay? So you see this C shape wrapping around in the brain. Okay? Here's the thalamus again. Okay? I mentioned the olfactory bulb earlier. Okay, this is where we're going to see a lot of smell processed and sending information into the brain. It's also connected with the olfactory nerve we introduced to the cranial nerves. Okay, so we're starting to integrate stuff together that we've talked about in the lecture. Okay, the hypothalamus is part of the diencephalon together with the thalamus. Okay, it's pointed in this little area up here, sort of this yellowish area. Okay, it's going to be ventral or underneath the thalamus. It's going to interact with the pituitary gland and control a lot of hormones, a lot of chemicals in the brain. It's a very old. Remember, the cortex is the newest part of the brain evolutionarily. As we get down to these areas, it's the older evolutionary areas that's shared with a lot of other species. The hypothalamus is very old. It's very much involved in regulating basic autonomic functions, fight or flight. Here's the way to think about remembering the hypothalamus. It regulates a lot of hormonal functions and chemical releases to govern the 4 F's of human behavior. Fight or flight, feeding, and reproduction. Talk to me after class if you need that broken down. Okay? So, that's our basic overview. Okay? Now, we talked about the thalamus, we talked about the hypothalamus. Okay, we talked about its interaction controlling the pituitary glands. Okay, and we talked about the thalamus relaying sensory information. Okay, we talked about our different planes of viewing the brain. Okay, we have these sort of slices this way or a slice this way. coronal slice or horizontal slice in blue. Okay, this is that horizontal slice. Okay, and this is helpful to look at different stuff. Okay, so this is our frontal anterior part of the brain. This is the posterior part of the brain, the occipital lobes back here. See how we can still see the gray matter and the white matter differentiating? Okay? Now, what we can see is that we can see these structures, these deeper dark areas of the thalamus. Up more anterior, we're going to see the basal ganglia. There's some white matter that runs through the basal ganglia. Okay? So it's helpful to view these structures that are otherwise sometimes difficult to view in other looks. Right? That's why we like convergent evidence. Right? When you think you're really discovered something, you're really onto some insight, you want to call somebody with a different point of view. Right? You have a left side version of the world. Before you really want to make a strong conclusion about some new theory you have, why don't you call one of your friends who has a really right side view of the world? Hopefully you have those friends and vice versa, because they're going to help us to see the world in the clear picture rather than our picture. Okay? Sometimes it's useful to see it in one view, but other views completely miss the picture. Okay? That's why we like all three. Okay, so we'll talk about these other things called ventricles too. We can kind of see these little holes in the brain, and we'll get to that just a little bit. This is that coronal view. Okay? Again, we can see our corpus callosum. Now see our basal ganglia again. See that here? Our basal ganglia. With the thalamus back here. Okay? Our basal ganglia, we have sort of the globus pallidus. Okay? All of our good stuff. But this this basal ganglia is sometimes visualized, like we just got done saying, better with a different angle of view. Here's a coronal view, and we can see that basal ganglia here. Okay, here we see the putamen. Here we see the caudate. Because remember, it has that tail. So you have that big, like the football, the ball of nuclei. That's the putamen. And then there's that tail that wrapped around. Remember that? That's sort of the ball here and the wrap around tail. Well, if you would cut this here, or here, you'd see the football, and then on top of it, after a little bit of space, you'd see the caudate tail with some white matter cutting through. Only because we're looking at it from a different point of view. Both are true, both are real, just different ways to visualize what's real and what's happening in the anatomy. Okay? Now with this view, we can see these other holes again, these ventricles. We'll talk about that, as I promised, again just in a few minutes. Here's our amygdala and that medial temporal lobe. You'll hear me talk a lot about the medial temporal lobe. I love it. Okay? The temporal lobe down here. See that Sylvian figure separating the parietal and the temporal lobes, and it comes into this, what we call the insula, and it has this huge fissure. Okay? Sulcus, huge sulcus, right? And it's not just this one, it really seeps in there. So you can really pull this apart. You pull these two apart. Follow? Okay. So we can do that. In the temporal lobe, it's the lateral temporal lobe. And in here you have the medial temporal lobe. That's where the hippocampus is. My favorite. Okay? The amygdala, very anterior, it is over here. So you get the hippocampus, amygdala, and that area. Okay? Now we talked about these sort of midbrain areas as well. Okay? So here we talk a lot about this cerebrum, the forebrain. Remember our forebrain here? Okay, let's break it down and get deeper. Okay? Go deeper down these layers. Okay? So we've already went from the forebrain broken down to the telencephalon and the diencephalon. Okay, we're going to break it down and work towards the midbrain. Okay? And we can look at these different features. Okay? We have these things called colliculi. Crazy words. I'm relying on you guys that this is not the first time you've seen the word colliculi. Because you did the homework and read beforehand. This is just review. Okay? There's two colliculi. Now, we're talking about basically the midbrain. That's going to be kind of, we're looking into the brain, it's going to be kind of here. We're going to have this sort of come into the spinal cord, right? So we're going to see that up along here, there's going to be these two little areas. This is where the colliculi are. And you'll see much better drawings. I'm just kind of giving an overview right now. Okay? And then we already introduced this idea of the substantia nigra. And we'll see these other areas called the reticular formations. And this other thing called the periaqueductal gray area. These are going to show up as just tiny little blurbs on little slices. Because the midbrain is actually pretty small. And sometimes the only way to visualize it are in little slices. Okay? So here's a better way of imaging this. Schematic. So we're looking at this midbrain area kind of over here. Okay? We have a pituitary gland, not part of the midbrain, but it's showing you some just orientation of where we are, the hypothalamus, the thalamus. Okay? Now, this is the pons. Okay? All part of our brain stem together with our midbrain. So we have an orientation. That's kind of what I drew here, that bulb, the pons. And we're looking at this sort of slice here, the midbrain. See that, we can see slices of it that include the superior colliculus and the inferior colliculus. It's going to be a little bump there and a bump there. Okay, that's that little bump there and there. You can also see the reticular formation. It's just a long column of nuclei. Okay, very evolutionarily old. Okay, controls just a lot of basic motor things, basic functions. Okay. These are really important for life-sustaining purposes, like we introduced earlier. Okay? So that midbrain is where the substantia nigra is, periaqueductal gray area, heavily involved in pain, perception and relief. Okay? And the reticular formations, these are columns of neurons. And we'll dive in a little bit deeper to the medulla and then, of course, the connections to the spinal cord. Medulla and pons, very, very old, evolutionarily basic life-sustaining functions. Respiration, heart rate. And of course, don't forget about our friend the cerebellum, even though most of the field oftentimes does. Okay? And if you think this is a lot of anatomy, you could have like a whole class on just the cerebellum and the anatomy. It's crazy. There's a whole lot more lobes of the cerebellum. We're not even going to get into it. It's crazy. But it's really cool. Anyways. So we introduced this idea of the pons and the medulla. Okay, so that medulla is transitioning to the spinal cord like we talked about. Essential processes, respiration, heart rate. Okay, some of the cranial nerves are coming in there. Okay, we've already talked about this. The pons, we already talked about that motor nuclei, some of the cranial nerves as well. And of course, we've talked about the cerebellum, motor control. People who have lesions of the of the cerebellum oftentimes have trouble coordinating their movement, and they walk in very disjointed ways. You might call cerebellar ataxia, A-T-A-X-I-A, not attack, but ataxia with an X. And because they're so disjointed in their motor coordination, they're oftentimes picked up by the cops during the day because they think they're stumbling around drunk. They're not if cerebellar ataxia or lesions of the cerebellum. Yes, ma'am. No, you can't use that as an excuse if you get picked up in the middle of the night. Sorry. It's a little bit different than cerebral palsy. It's actually a lot different than cerebral palsy because remember, cerebral is not saying cerebellum. Cerebral is different than the cerebellum. So completely different anatomy. Even though it sounds like they're really similar words, they're actually quite different. Okay. So this is another way to look at that from a ventral point of view, looking underneath the brain, the same way I've shown this for a couple slides over the course of a couple lectures. Here's our spinal cord coming in to the brain stem, the pons. We can see the inferior and superior colliculi a little bit higher above here. Okay, we looked at this rotated when we looked at the cranial nerves earlier today. Okay, we have the olfactory bulbs, okay, where some of the olfactory nerves are coming in. We'll see this these decussation here, this cross, cuts in actually from the ocular nerves. Okay, so the vision coming in. You have the temporal lobe, the cerebellum, frontal cortex. Okay, another way of looking at things. Okay, All right, let's see. Okay, so we already talked a little bit about these ideas of the meninges in the context of the spinal cord. However, we also have the meninges that we can see in the context, really, of the brain. Okay, so again, we've introduced the idea that there's 3 layers, that is, the dura, the pia, and the arachnoid space. Okay, so the dura is the tough the most outer layer, the most superficial outside. So the brain, so let's say you're going to do surgery, you got to cut open the skull, but you don't get the brain because it was designed so that if you crack your skull open, you still have protection from your brain from the dirt that's going to get in from the ground that you fell on, the rock that cracked open your skull. We got to protect that brain. You need it. Okay, it's very, very sensitive thing. Chemically, cellularly. We got to have layers around it to protect it. So you have the outermost layer has to be tough. Right? And so that is what the dura is. Anybody here speak Latin? Okay. What's Latin for tough mother? Huh? Yeah. Tough mother. You can imagine what they were thinking of characterizing it as. Okay? Pia mater is a little bit closer in. Doesn't have to be so tough. Okay? It's more sensitive. Alright? The arachnoid member, basically the arachnoid space. It's that area that's between the two. And that's going to be filled with cerebrospinal fluid. Yes?
Pia mater also means tender mother.
Yes, it's more sensitive. It's a tender mother in Latin. And so these are basically contain cerebrospinal fluid. Fluid that's kind of helping to balance the brain. So the brain actually rests in a little bit of fluid, particularly in the ventricles that we'll talk about next. Okay? And it allows it to float a little bit. And so it can move a little bit within the cranium, which is also helpful. It's also one of the ways that you get concussions, is it floats within this little fluid, and when you get a fast acceleration, the brain might rush forward and hit the skull. Because it's not fixed, it's floating in fluid. You might get a little bruise or a little damage based on that accelerated impact of the inertia. Okay? So these meninges you might recognize as being sources of diseases that you might have heard of. Meningitis. Itis means inflammation. Inflammation of the meninges. They get sick. They get attacked. They get inflamed. Spinal meningitis. Inflammation of the meninges are on the spine. Okay? You can also have meningiomas. Omas, meaning tumors. We have tumors in the meninges. Okay? Any of you guys are just really into like hardcore sick images, just Google that stuff on Google Image. You can see a lot of really interesting medical stuff. Yeah, it's pretty crazy. So we have that stuff. Okay. Now I promised to return to the ventricles. The ventricles are basically fluid-filled chambers. They're these sort of thought of as empty space, but it's not empty. It's got fluid in them and stuff like that. Okay? Filled with cerebral spinal fluid. There's several ventricles. It's a system. They're numbered 1, 2, 3, 4. Okay? Now, inside the ventricles, the thing that actually makes the cerebral spinal fluid is called the choroid plexus. Okay? Now, the lateral ventricles are the big sort of horn-looking ones in both of the left and right cerebral hemispheres. Okay? That's these little triangle areas that I've drawn here on this brain are showing that coronal slice through one of those horns that wrap around in that same C-shaped structure. We also have a third and a fourth ventricle, okay, that go down the midline and then exits to circulate over the brain and spinal cord. Okay, so this is an image that shows a little bit better what it is that we're talking about. Okay, here's a lateral or a sagittal view. We have that C-shaped lateral ventricles It comes down through the third and the fourth ventricle. Okay? I can take that cerebral spinal fluid produced by the choroid plexus and transmit it down to the spinal cord. This is looking down from that top view, or an axial view. Yes, Jim?
Yeah, it's going to sound silly, but is there a second ventricle? You got a lateral. You got a 1 and a 2. With 1, 3, and 4.
1, 2.
Oh, so they're each side named number then?
I mean, they're generally just called the lateral ventricles.
Okay.
But since there's
So they call that 1, 2 then. Okay.
since there's 2 hemispheres, there's one.
All right.
Uno, dos.
Okay. All right. But there's only 1, 3rd, and 4th then, right?
Yeah, because they tend to drop right down the middle.
Okay. All right. Got it. All right. That makes sense to me then.
Yeah. So they're gonna come down. This is another way of looking at it. This red stuff is the choroid plexus producing this fluid. Okay? Sometimes you can have developmental diseases where it's producing too much, or it's not exiting down in the spinal cord, and it can be fatal. And what surgeons will do is put a shunt in to help drain it to the infants, which means open up, drill a little hole in the back of the skull, and let it drain out. It can be life-saving. Yes, Jeff?
So the red is the choroid plexus?
Choroid plexus.
Or that area there?
Yeah.
Okay. All right. And that's, in other words, not all of it, it's just that area that's producing the fluid.
Yep.
Okay. Thank you.
Yep. So you might recognize this because this is what Da Vinci took the mold of the ox that we talked about in history. Yeah? Okay. Cool, right?
No, I didn't get that one.
Huh?
I didn't get that. Which was that?
So remember we looked in the history lecture about Da Vinci started off, there was a slide, you might remember it. It started off with a sideways view of a guy with a nose, lips, chin, right? This is, and it came in, and initially the view, historically, right, and this would be like, this was the view of the ventricles.
Oh, so that's what he did. That was a...
No, this is what he drew at first, and this is the predominating view, and then he said, well, why don't I take an actual mold of this anatomically, because he was a Renaissance man, and he was interested in anatomy and everything like that. So he took paraffin wax, filled into basically a dead oxen's brain, let it mold, then peeled the brain back, and actually was able to see the full shape of the ventricles, and he found instead that, they basically looked like that structure that we're looking at here. There's much better slides and pictures of that. Yes?
Are the lateral, or are all the ventricles just in charge of the fluid? Is that all they do? Or
For the most part.
Yeah, okay.
And then, of course, sort of we also have the cerebral aqueduct too, sort of where they're connecting. Okay, so you have the third ventricle, the cerebral aqueduct coming down and connecting with the fourth. Okay, so let's talk about a couple other things as we move along. Great progress, by the way, today, guys. Is everybody able to hold on, even if only by like a little fingertip or two? You still feel like we're being dragged along? We still got it. Okay, awesome. Great job. Keep it up. Keep holding on. We're halfway there. I'm living on a prayer. Okay. I'm from Jersey. I gotta get Bon Jovi in there a little bit. But the idea here is that we have a little bit more to go. So we can also talk about the vascular system of the brain. If the brain doesn't get oxygen, it dies. Okay, that's called hypoxia. You're under a lack of oxygen. Now, the way it gets oxygen is the delivery system is blood, right? It's true for all sorts of stuff. And so we need to oxygenate the brain, and we have blood vessels that accomplish that. So we have vascularization. You remember we've introduced this idea when we talked about Sir Thomas Willis earlier in the history lectures too, right? He categorized this very elegant vascularization system, and we remember we have the circle of Willis, which was several of those arteries, okay? Now, it's basically what's involved in oxygenating the brain, and we can see pathologies and deficits that happen when we disrupt the system. Stroke is a good example of this, okay? You can have different kinds of strokes, okay? Kinds of strokes where the arteries are blocked, or kinds of strokes where they're ruptured, like an aneurysm, okay? Either way, the area of the brain that is supplied oxygen by those arteries is injured. It dies, because that area doesn't get oxygen, and those cells go, cells get damaged. If those areas were areas involved in language, we get language impairments, and motor activity, etc., etc. Okay, that's kind of what's shown here. You have a blockage of an artery in this area that might be innervated or receive the oxygen from that blockage is going to have trouble. Okay? You can also have a rupture. Same idea. You're going to have areas damaged by virtue of that. Okay? Now we can have several different types of arteries that we use our anatomical orientation terms to categorize. You can have a posterior cerebral artery, anterior cerebral, a middle cerebral artery, right? Which categorizes what part of the brain are those arteries projecting to. Middle, anterior, posterior. Make sense? So I went over a lot of that. Okay? Okay, so if you have like a posterior cerebral artery stroke, that artery often innervates a lot of the temporal lobe. Okay? So that's very useful for my research, as tragic as it is for the patients, because the posterior cerebral artery innervates the temporal lobe, which is where the hippocampus is, which means that you can often get memory and amnesia deficits from a PCA stroke. And then we can go in and we can test those amnestic deficits and understand that type of amnesia and the role that those areas play by understanding the deficits that are there when those areas are damaged. Make sense? Okay. Another element of the vascularization system that's important to note is the idea of the blood-brain barrier. Okay, so these blood vessels and capillaries that go all over the brain, okay, they're involved in carrying not just oxygen, but nutrients and nourishment to the brain, and also cleaning out the detritus and bringing stuff back, get rid of stuff. And so it is not just a tube, like a hose, bringing blood, because how does it get out then? It's actually a mesh membrane, like mesh shorts. How many of you guys would, if you didn't have running water at your house and you needed to go down to the well in the river and get a bucket of water and bring it home, old school style, how many of you guys, instead of, if you didn't have a bucket, would use your mesh shorts to collect water? Tie them up in a little bag and carry them over? No. Terrible idea, right? Because it's going to leak. Same thing here. This is a blood vessel. It's a coronal cut through it. But what we can see is that there are these mesh. It's mesh. These are oftentimes the astrocytic foot. Right? Remember we talked about that, making that connection. But the mesh elements of the membranes on the blood vessels are held together really, really tight. Oftentimes because of pressure in the atmosphere. It just keeps everything tight. Certain gases, certain chemicals, certain nutrients are molecularly small enough to pass through those holes in the mesh. And that's how we can deliver drugs and nutrients and oxygen and food into the brain. It leaks out of the blood vessels. But the blood-brain barrier refers to the idea that there's a barrier that will not let molecules of a certain size pass. It's a big problem in the pharmaceutical industry. When we want to make medicines to help people and fix problems in the brain like depression, anxiety, Parkinson's, Alzheimer's, the drugs that we want to deliver are too big to pass through the blood-brain barrier. Wah, wah. Can't do it. So what you have to do is get creative and develop precursors to drugs, chemicals that the brain can synthesize into the drug, oftentimes. An example of that is L-DOPA, used to create and synthesize dopamine to treat Parkinson's patients. If you want to see a really cool video about that, or movie, check out the Robin Williams movie called Awakenings. It explains that in a clinical context of a doctor who discovered that. It's really cool. Robert De Niro's in it, Robin Williams, and it's a great movie. So you have this blood-brain barrier held together by pressure. So one example of a dysfunction of this system is when you mountain climb. I climbed Kilimanjaro when I graduated college. 2005. I went from the Jersey Shore at sea level to 19,350 feet in 5 days. I was an idiot. What happened is I got high altitude cerebral edema and pulmonary edema, which meant I nearly died. I was pretty much on a deathbed. And the reason of what happened pathologically was because you went so high in the atmosphere, the pressure of air gets lighter because the air is thinner. So it's not crunched down on the blood vessels so much, it's a little bit more relaxed. Those holes in the mesh are no longer pressurized to be real tight. They've now opened up a little bit, and all that stuff we like to keep inside our blood vessels, it just leaked out. Fills your brain with all this extra fluid, you have edema, and you have lots of deadly and terminal things happen because they're leaking through the blood-brain barrier that's otherwise held together very nicely. Okay? So that's kind of the idea there. Indulge me just a moment. What do we have, about 5 minutes left? Two whole minutes. I'm sorry?
150?
150?
Yeah, it's 148.
148, okay, we got time. All right, let's do this thing. Come on, you're paying for this. I don't want to shortchange you, all right? Okay. So let's talk a little bit, real quick, about some of the elements in Chapter 4 that I put on the syllabus, okay? Some of the transmitters in pharmacology. So what I'll say is when you study that chapter, okay, you don't have to study basically the back half about the applications of transmitters like drugs, drugs of abuse. Just understand the systems, okay, of the transmitters that are there, okay? You don't have to worry about, say, figure 4 right now, but I wanted to introduce the idea that you have different receptors for different transmitters, okay? What I want to show you are basically this idea. Know that we have GABA, glutamate, okay? We have acetylcholine, dopamine and serotonin, and some gaseous neurotransmitters like nitric oxide that work as retrograde signalers going backwards across the synapse, okay? So know a little bit about acetylcholine, okay? We'll talk about that hopefully a little bit next class. We have glutamate, okay? It's excitatory. We have GABA, which is inhibitory, okay? And serotonin and dopamine also play a role with norepinephrine. And these are some of the systems, okay? So know these elements. That dopamine is often associated with the nucleus accumbens, okay? Noradrenergic systems, okay? With basically the locus ceruleus. Serotonin with the raphe nucleus, okay? Just know basically some of those nuclei. We'll start off next class addressing some of that and sort of just visiting this in more detail. But just know these areas where they project through, okay? Thank you for your attention, guys. You did really good today. We covered a ton of stuff. You did really, really good.
You didn't go to like the base camp and stay for a while to let your body regulate?
Not a whole lot. I mean, I had a few base camps, but it was functionally useless. Yeah, well, 22 years old, pretty, you know.
Yeah. I was gonna say without you didn't give your body time to.
Nah.
Oh wow.
Yeah.
How long were you in care?
Just a day or two. I mean, they saved my life. It was very gracious.
Somebody bring you down? Or
Yeah.
Someone go get you.
No, I can't. I mean, I got down to about 12,000 on my own, and then basically got rushed off the last 12 with a stretcher by some amazing porters who basically saved my life.